Why do some people seek danger? Others avoid conflict. Some animals cooperate while others fight. Is it choice? Is it experience? Or is something deeper, molecular, invisible, shaping how brains behave? What if behavior isn't just learned, but built? Welcome back to my channel, where we unravel life's biggest mysteries written in your DNA. Today, we're diving into one of the most complex questions in biology and neuroscience. Are genetic and biochemical pathways central to behavior in humans and animals? And if they are, how do genes, brains, and environments work together to shape who we are and how we act? There is no single gene for aggression. No lone gene for curiosity, no switch labeled personality. Behavior is polygenic, shaped by many genes acting together in networks. Some of the most important genes involved in behavior regulate neurotransmitter systems like dopamine and serotonin, stress and hormone signaling, neural development, and synaptic connectivity. Across both humans and animals, researchers repeatedly find shared genes influencing behavioral regulation and resilience. Genes like OPRM1, MPY, CASNA1C, DCC, and FKBP5. These genes don't dictate behavior directly. They influence how neurons communicate, how stress is processed, and how brain circuits respond to experience. And crucially, their effects depend on context. Genes don't act alone. Genes shape behavior by shaping biochemistry. Neurotransmitters like dopamine and serotonin don't just affect mood. They regulate motivation, like reward, impulse control, learning, social interaction. But behavior doesn't begin at neurotransmitters. It begins much earlier, during brain development. Developmental signaling pathways like WN-beta-catenin, Notch, sonic hedgehog guide how neurons are born, migrate, and connect. Even biochemical processes like protein glycosylation and sulfation quietly influence how neural proteins fold, signal, and stabilize, with lasting effects on cognition and behavior. By the time behavior appears, the molecular groundwork has already been laid. Here's where things get really powerful. Behavioral genes don't work independently. They're controlled by gene regulatory networks, or GRNs. Think of GRNs as molecular orchestras, coordinating the expression of hundreds of genes in response to stress, social interaction, environmental change, learning in seconds. These networks interact directly with neural circuits across time and space. Studies in zebrafish and insects show how tiny genetic changes can alter sensory sensitivity, circuit wiring, startle responses, and chemosensory-driven behavior. A small shift in gene regulation can reshape an entire behavioral output. Behavior emerges not from single genes, but from systems talking to systems. Genes matter, but environment decides how they matter. Gene-environment interactions shape behavior through epigenetic modifications, experience-dependent brain plasticity, microbiome brain signaling, stress, nutrition, toxins, caregiving, social support, all can alter gene expression without changing DNA sequence. During critical developmental windows, prenatal life and early childhood, these interactions can permanently shape brain circuitry. Even the gut microbiome can modify behavior by altering brain metabolites and gene expression in regions like the prefrontal cortex. Genes load the gun. Environment decides when or if it fires. Many behavioral pathways are deeply conserved across species. That's why animal models are so powerful. They reveal causal gene-brain behavior links, they allow controlled genetic manipulation, they inform psychiatric and neurological research. But translation isn't automatic. Species differ in gene regulation, pathway timing, and circuit architecture. Understanding behavior requires both comparative insight and species-specific caution. Behavior is not written by genes alone, and it's not written by environment alone. It emerges from a constant molecular conversation between DNA, brain chemistry, neural circuits, and lived experience. Understanding behavior means understanding systems, not shortcuts. And the more we uncover these pathways, the clearer it becomes. Who we are is shaped long before we act, but never beyond change. What other hidden genetic systems should we uncover next? Until next time, see you soon.